Hello, students. Let me ask you a curious question. How do you think nearly 2 meters of DNA can be packed into a cell nucleus that is just a few micrometers wide? The answer is one of biology's most fascinating packaging miracles, chromatin. Chromatin is the material inside the nucleus that forms the basis of chromosomes. It's not just DNA, but a carefully designed partnership between DNA and proteins, mainly histones. About 40% is DNA and the remaining 60% is protein. In its relaxed form, chromatin allows the cell to function normally, but when the cell prepares to divide, the chromatin coils up tightly into distinct chromosomes. Isn't that an incredible transformation? To understand chromatin, let's first look at its heart, DNA, deoxyribonucleic acid. The shape of DNA was famously worked out by Watson and Crick in 1953 based on Rosalind Franklin's X-ray diffraction studies. They described DNA as a double helix, two complementary strands twisted around each other like a spiral staircase. Each strand is made of repeating units called nucleotides. And what's a nucleotide? It's made up of three parts: a phosphate group, a sugar called deoxyribose, and a nitrogenous base. This simple repeating design creates one of the most extraordinary molecules of life, capable of storing and transmitting all our hereditary information. Now let's zoom into the rungs of this DNA ladder. These rungs are formed by nitrogenous bases joined together by hydrogen bonds. There are four bases: adenine, guanine, cytosine, and thymine. But here's the key rule: adenine always pairs with thymine, joined by two hydrogen bonds, while guanine always pairs with cytosine, joined by three hydrogen bonds. We write this as A equals T and G triple bond C. This strict pairing rule ensures accurate replication of DNA every time a cell divides. Just imagine, every cell in your body relies on these precise pairings to pass on life's instructions without error. Now comes the clever packaging. DNA on its own would be too long and fragile to function. This is where histone proteins step in. Histones act like spools, and DNA winds around them. Together they form structural units called nucleosomes. Each nucleosome is made of DNA wrapped around a core of 8 histone proteins, also known as the histone octamer. Picture this like thread wound neatly around a spool, preventing tangling and making storage easy. This is how cells manage to organize such massive molecules into compact, functional units. But the story doesn't stop there. Nucleosomes coil further into thicker chromatin fibers. And these fibers then undergo supercoiling, much like the coils of a telephone cord, to become even more compact. This hierarchical arrangement ensures that DNA not only fits inside the tiny nucleus but is also accessible when needed for transcription. If you were to look under an electron microscope, you would see these chromatin fibers as delicate, thread-like structures filling the nucleus. Now let's connect this to cell division. During the normal functioning of a cell, chromatin is kept loose and open so that genes can be read and proteins can be made. But when a cell is preparing to divide, chromatin undergoes a dramatic change. It coils and condenses further, transforming into thick, visible chromosomes. This ensures that the DNA is copied and distributed evenly into the daughter cells. So the journey goes like this: DNA winds into nucleosomes, nucleosomes form chromatin fibers, fibers coil into chromatids, and finally, chromatids form chromosomes. Isn't that an elegant sequence? So let's wrap this up. Chromatin is the DNA protein complex that packages the long DNA strands into a compact, functional form inside the nucleus. DNA itself is made of nucleotides with complementary base pairing, adenine with thymine, and guanine with cytosine. Histone proteins help coil this DNA into nucleosomes, which then fold further into chromatin fibers and ultimately form chromosomes during cell division. Without chromatin, it would be impossible to fit 2 meters of DNA into a tiny nucleus or to regulate which genes are active at the right time. Isn't it amazing how nature has engineered such a perfect system of packaging and accessibility? Every single cell in your body relies on chromatin to protect its DNA while still keeping it ready for action. Remember, this beautiful balance of order and flexibility is what makes life possible. Until next time, keep wondering about these marvels of biology, because every answer uncovers yet another layer of life's incredible design.